Hello and welcome to With My Plant. Today we are going to understand the ABA signaling in stomatal movement. Here the movement meaning the closing and opening of stomata. So when this stomata is being closed. So this is the first question arising here. So when the plant is facing a stress, it will try to store its water, its absorbed water, and for that it needs to close its stomata to reduce the transpiration. So what is happening when a root is facing root is the first organ which is sensing the water stress. So when the root is sensing the water stress, it will produce, the plastid of the root cells are producing ABA, and through xylem this ABA is going to leaf, and in the apoplastic region of leaf uh, it is accumulated. Okay, so this accumulated ABA will now start signaling in the guard cell. So in the guard cell, what is happening, this is the picture of a guard cell and this is the stomatal pore. So in the guard cells, what is happening, there are receptor present in the cytosolic region of guard cell, and they are receiving the ABA. So these are the receptor RCAR, PYR and PYL. Okay, after receiving this ABA, this ABA receptor complex is now forming another complex with PP2C, which is protein phosphatase 2C. Okay, so this is a negative regulator of this ABA signaling, and this is basically this inhibition of this complex through ABA receptor complex is activating the OST1. OST1 is basically a kinase enzyme which is through phosphorylation activating RBOH. RBOH is a NADPH oxidase enzyme which is basically producing reactive oxygen species. Here we can see the reactive oxygen species produced by this RBOH. Now this reactive oxygen species is helping the activating the calcium inwardly rectifying channels, which is basically responsible for the accumulation of calcium in cytosolic region of guard cell. Okay, so by accumulation of this calcium, it is again activating another kinase enzymes, which CPK enzyme basically, and these kinases are regulating the anionic outwardly rectifying channels. The anion can be chlorine, can be any anion. Okay, so this is basically reducing the concentration of anion in the cytosol and increasing the concentration of calcium ion in the cytosol. And this anionic complex or anionic outwardly rectifying anionic channels, by reducing this anion concentration, basically again regulating the calcium outwardly rectifying channels, which is controlling the concentration of potassium as well. So potassium concentration is decreasing, anion concentration is also decreasing, and calcium concentration is increasing in the cytosol. In the other hand, what is happening in the vacuole, the calcium outwardly rectifying channels are being activated, and also the anion outwardly activating channels and potassium and outwardly activating channels are being activated, which is basically increasing the concentration of calcium in the cytosol. But potassium and anions, as the outwardly rectifying channels of plasma membrane is also activated, so these are being reduced in the cytosolic region. So what is happening, calcium is increasing, potassium and anions are decreasing. Okay, so what is happening again, and because of this, what is happening, the osmotic pressure is being reduced, and stomata turgidity is lessening. That is how the stomata is being closed. So in this part here we can see it is indicating the inhibition of influx. Okay, so when ABA is there, it is basically uh, enhancing the calcium concentration, which is again uh, uh, regulating the inwardly rectifying potassium channels and stopping the potassium concentration or reducing the potassium concentration. So it is acting in two ways. So increase in calcium is regulating two to three things. First, calcium entering is blocked and calcium exit is enhanced and anion exit is enhanced. Okay, so what is happening? All potassium is out, anion is out, only calcium is in. So this is how the calcium concentration is increasing in the cytosol of guard cell, and that is because of this ABA. The root cause is the ABA. So this is how the stomatal closing is being processed. In the second slide, I have given a simple note of this. You can read and prepare a note by your own self, which will help you in the exam. Thank you. This is all for today's topic.